All right. Yeah, so today's last session is about tracking neurons across days, as Laura just mentioned. We're a bit ahead of schedule, but I'll try to keep it short anyway. So why would we want to track neurons? So here is an example of Julie, who you saw earlier today, who did an experiment when she was in her lab where she recorded neurons in the basal ganglia while mice were learning this task where they had to bring a lateral stimulus to the center. And what she observed is that neurons, on average, in the basal ganglia fired more when the central stimulus appeared, which is associated with a reward, and only then started increasing the response to the action leading to the reward. But is this really true? So to really know if this is true for all neurons, we need to record neurons over time and track them over time. So for example, if we want to study decision-making, learning, memory, or aging, we really need to cover this entire scope of time scales. And for a long time, people have used two-photon imaging to do this. So as you know, for two-photon imaging, you can really see the cells over time. And if you keep going back to the same field of view, if you're lucky enough, you can find the same cells quite clearly. But of course, two-photon imaging doesn't reach the time resolution that Neuropixels or generally electrophysiology can. So this is where Neuropixels and other electrophysiological tools come in. So as you know, if you acutely insert these probes, you can reach all the way from milliseconds to a few minutes to an hour. However, we've seen that you can chronically implant these probes and really extend the time scale that you can record from in the same mice across behaviors. But of course, that doesn't completely solve the problem because we cannot really see those cells as we can in two-photon imaging. So how do we know which cells in one recording are the same in another recording? So as you know, when we spike sort, we typically assign the spikes across a recording to specific neurons. So if we have neurons here sitting next to a schematic of a probe, we ask which of these spikes belong to which of the neurons. And there's many methods to do this. One example that has been mentioned a lot is kilosort, but there's others. So if we now have a chronically implanted probe, we can just pretend that this was actually one recording and concatenate them together and then run the spike sorter. And hopefully the spike sorter will assign the spikes across the two recordings to specific units. So we don't have to bother with tracking neurons in different recordings. But now we can actually record for many weeks from the same implant. So this really scales up the data we have, and this is just no longer scalable to use this method. So this is why we came up with a method to track neurons across days more flexibly and more scalable, which consists of three parts. We call it unit match. The first part is pre-processing, the second part is the matching, and the last part is the tracking. And I'll go into more detail. So for the pre-processing, what we're doing is we're spike sorting individual recording sessions using kilosort in our case, but you can use whatever you want really. So every single session has a separate spike sorting done. We then use curation, so we use bombcell, but again, there's other methods out there. And we ask for all the good units, what was their waveform averaged across the first half of the session and the second half of the session. And we extract all these waveforms. So just to make clear that this is just one neuron, it's the average waveform on every single channel in that recording for the first half and the second half of the recording. So now we want to compare these waveforms and we want to ask which of the neurons have similar enough waveforms to be called the same. So for example, if this was our original neuron, we can look at the waveforms for the closest neighbor and ask, is this similar enough or not? And to another neuron in another recording and ask again, is this more similar to this one or this one? Well, my eyes are not good enough to really parse this information. So instead, we define some parameters to extract from these spatiotemporal waveforms. So first, we're looking at the maximum site where we had the maximum amplitude for each neuron, and then plot the amplitude as a function of distance to this maximum recording site. And we can extract a spatial decay parameter from this information. We can also look at the weighted average temporal waveform. So we can extract just the average waveform, what it looks like, and the amplitude also weighted across the the channels up to a certain distance. Because Neuropixels has very high density recording sites, we can use this to follow the electrical current in time within a single waveform, so basically within a millisecond. So we know that in neurons, the electrical current travels a little bit within the neuron. So if you have enough density, you can actually plot the weighted position based on the amplitude at a given time from, in this case, minus 0.2 to 0.5 milliseconds from the start of the waveform. So from this trajectory, we can again extract some parameters that might be useful to tell us which neurons are the same. So once we've extracted these parameters, we can compare them across all our data. So here is the closest neighbor again overlaid on top of the original neuron. And as you can see now, the waveforms are pretty different, both in the temporal and the spatial domain. And indeed, if we look at these individual scores, which I showed here again, we see that only the centroid score, which is basically the distance between the two cell bodies, is very high, but all the other scores are really low, leading to a relatively low total score or similarity score. If we do this for all the neurons on day one, for the first half of their recording versus the second half of their recording, we can see that the neurons are very similar to themselves across the two halves. So that's the main diagonal, which is great. And then outside of this diagonal, the scores are overall lower. And we see the same for day two. So we can now plot this as a function for neighbors in blue. This is 50 micron around the neurons, and the same neurons in the two halves. And we find that the same neurons have generally higher total scores than neighbors, such that we can find a threshold where these two distributions are really different. If we now look at the best match across days, we can see that there's a waveform that is actually very similar both in temporal and spatial domain. And indeed, all the extracted parameters are relatively high, leading to a higher total score. And indeed, if we look across the 2 days, we see a second diagonal. This is sorted based on depth, which indicates that there are pairs of neurons across the 2 days with very high total scores. So now we can plot this full distribution again, and we can find that we can apply the same threshold to this distribution and label these ones that pass this threshold as putative matches. We can now use these thresholds, so everything larger than the threshold and everything lower than the threshold, and go back to the individual scores and look at the probability of these scores happening for these two groups. And if we then put them in a naive Bayes classifier, we basically clean up this matrix from earlier, really binarizing the data almost to having either very high match probability, so high probability of two neurons being the same, and very low probability in white of two neurons not being the same. And we indeed find that this main diagonal is back, indicating that we are able to find the neurons with themselves between the two halves of a recording, which is a good sanity check. And we can find the neurons in another day, so across days, also with high probabilities. So now plotting the same data, but as a function of probability, we indeed find this same neurons with high probabilities, neighbors with low probabilities, and across days, a mix of the two. And now these are the ones that we're actually interested in. So now we can say, okay, neurons with very high probabilities are more likely to be the same neuron, so we can assign them the same identity across 2 days of recordings. But how do we know that these matches are valid? So one way is to look at independent parameters. And in our case, because we have not looked at any functional activity yet, we can use the functional activity to validate our findings or our matching. So in this case, we're looking at 3 times the exact same plot. This is the black neuron again, and we see its interspike interval distribution. So this is the proportional spikes at a specific interval between the spikes. And if we now compare that to the second half of the same recording, we see that they are very correlated. And in fact, if we do this for all the neurons within the same day, we find very high correlations. If we now do the same for another neuron in the same day, we see that these distributions are very dissimilar. So this leads to very low correlations. And these two distributions across all the neurons are so different that we can actually use it to define whether or not a neuron was the same neuron based on these correlations. And this is a receiver operant characteristic curve with an area under the curve very close to 1, showing that this correlation is very telling, telling whether or not it was the same neuron. If we now look at matches across days, we see again very high correlations, almost as good, or in this case as good as the same units within days. And indeed, the AUC value is also close to 1. So this means two things. Our algorithm is very good at detecting the matches across days, but also that these functional scores are quite stable across days. If one of the two is not true, this value would go towards 0.5. So this is an example mice. So this was just 2 days, 2 recordings across 2 days, but we can do this with different spacings between the recordings. So we've done it all the way up to 200 days. This is a logarithmic scale. And we find that this AUC value is very stable. So again, these the matching is done quite good, and also these functional scores don't really change much over time. And this was true for basically all the mice we had. There was a bit of variability, but it was well above 0.5. But of course, we don't want to just do this for 2 recordings, because then we can just concatenate and run kilosort, but we want to do this across a whole data set. So here you see 22 recordings. You see the main diagonal of the neurons with themselves, and you see these off diagonals for every pair of recordings, showing all the neurons that were shared across these recordings. So how do we now use this information to find the unique neurons across the recordings? So to do that, we designed a tracking algorithm where first all the probabilities are sorted from highest to lowest. So we're starting at the highest probability and say, well, this is very high, so this is going to be the same neuron. We then go to the second highest probability and again say, well, again, very high. So now we know across the 3 days, this is the same neuron. But now we're encountering a kind of a conflict situation, because this is higher than 0.5, so it could be the same neuron. But with this group, it also has a case where it's actually a low probability. And we say, well, in this case, this one blocks this neuron to be added to the group. So we now have 2 neurons, one black and one blue identity. So using this tracking algorithm and unit match, we are able to find neurons back in the past or in the future. And we can even find neurons that if they're here on a certain day, they might have appeared quite late, or they even might disappear for 1 day and come back again, which is something that can happen with electrophysiology. And overall, it's quite good at detecting the same neurons. So it can handle disappearing and reappearing. So how many neurons can we find? This is always a bit tricky because it depends a lot on the quality of the recording. But this is just all our recordings we had at that time so far. So here you can see the number of data sets going into this graph. Even on the same day, you only see 55% of the neurons across 2 recordings. And this could just be because neurons might be less or more active, may not be picked up by kilosort anymore because they don't spike enough, etc. If we now ask, okay, how many of these neurons did we find in the past? It's up to 20%, all the way up to 200 days. So that's actually quite impressive if you think about it. If you now ask, how many do I find in the future? It's going down a bit more towards 10%, which suggests that overall, the number of neurons we can record from over long periods of time may decrease based on probably some buildup around the probe. Again, we find that these AUC values are well above 0.5. They do decline a little bit. This could be either because the functional activity actually changes or because the method gets slightly worse. Of course, we don't know that, but because it's well above 0.5, we do think it's the matching is done correctly. The same was true for visual response plasticity in this case. So going back to this example, is this true for all neurons? We have here 3 individual neurons matched across these recordings, and we do find that some neurons increase response to just one stimulus, while other neurons increase first to one and then to the other. So in summary, no, we really do need a single neuron tracking to really understand what's happening during learning. I just want to highlight that there's other methods. So there's methods also by Augustine Juan and Jennifer Colonel, which is using Earth movers distance that you just heard about. There's a talk about this in the 2023 version. I'll share the slides so you will be able to access that there. And we also have been working on further optimizing tools. This is in collaboration again with Céliane Bimbar, but also supervising two very motivated and good researchers, Wen Tao and Suyash, who've been developing a deep unit match, which is basically using a deep neural network. I don't have a lot of time, so I'll go over it very quickly. Basically, what it does is instead of me designing parameters, which may or may not be the right parameters, is learning a low-dimensional representation of these waveform snippets by having an autoencoder trained. We then optimize this encoder further by asking it to optimize between the two halves of a recording using backpropagation with contrastive learning. And then once this is a fine-tuned encoder, we can use it to inference and basically assign similarity scores of neurons across recordings. Because this is similar to the total score I've told you about earlier, we can just plug it back into the unit match pipeline and assign probabilities, do the tracking as we have done before. And yes, indeed, unfortunately, AI is better at than me at finding the right parameters, because overall deep unit match seems to be better in terms of functional validation than unit match. We worked hard on getting this demo ready for today. So if you're interested, yeah, go check the demo for deep unit match. That was it. I would like to thank again my collaborators, especially Céliane, Suyash, and Wen Tao, and the useful links will be on the Slack channel. Thank you.